talks too much. Thank you for the invitation to talk to this group. Some of you I've spoken with before about these malaria research efforts and some not. I'm going to present some, quote, old data, but also, in addition to published work, I'm going to talk about some work that we have really exciting data that's not published yet. Now, as the title shows here, I'm talking about endothelial dysfunction in malaria. And all of this work started some time ago. It started here at Duke when people who were working with me decided to get into the malaria business more with the work and the help of Dr. John Hamilton in infectious disease, Dr. Nick Anstey here. Not sure that arrow shows up at all. On the big screen, maybe a little bit on the little one. Dr. Hamilton arranged for Dr. Anstey and his wife to travel to Tanzania to study malaria. And this study was done with Dr. Esther Maikambo, who is this lady here, Maikambo. And she was a malaria expert, a pediatrician. She had worked for years in malaria. And she helped Dr. Anstey and his wife see patients on this protocol. They were all children, and study what I'm talking about today. The work over the years, which has gone on for several years, has taken place in the United States, in Tanzania, Australia, Indonesia, Malaysia, Bangladesh, and some other places too. I'm based here in Durham. Dr. Granger, who's one of the main collaborators, used to be at Duke in infectious disease. He's originally from Utah. He moved back to Utah, and he's now a professor of medicine there at the university. And I'll mention a few other people who've been involved in this. Now, many of you know because you're global health specialists, and if you don't know, you definitely need to learn about malaria because it's so important worldwide. In hematology and oncology and nephrology and pulmonary, we see some diseases where we get so excited because there may be 1,000 cases a year or 10,000 cases a year. In this disease, we're talking about millions of cases a year. And in addition to the incidence, the severity and the number of deaths is huge. The last counting that I think is accurate was over 400,000, and most of the deaths are in children and in pregnant women. And there's some good background to explain that, but I won't go into that today. And despite best treatments, a large fraction of these people with malaria go ahead and die. If they get into a hospital, if they get the best treatment, many of them still die. If this is your 2-year-old daughter or son, or an 8-year-old daughter or son, and that person dies, it is a true tragedy. We've had some people from the Duke community and from patients here in the past who've gone over as missionaries, some people from Southern Virginia, some people from Duke. One of the people, postdocs who worked with me, went to Gabon, didn't take his medicine prophylaxis properly. He developed severe malaria and was hospitalized. Other people developed severe malaria when they had no immunity, and they died. So there have been some young college students who died from this too. In addition to the death and the acute severity of needing to be in the hospital, it has a dramatic effect on the population's wellness and productivity. These people are very sick, they can't work, and it just has a very large national impact. Most of the infectious diseases benefit from a vaccine. We take this for granted. It is certain that in malaria, there is not a good vaccine available. Some are being developed, and there's one about to be FDA approved. It has the ability to protect against malaria for maybe 30 or 40% of patients. So it's not like you're getting your flu shot or not like getting a tetanus shot or something like that. It's not a good vaccine, and people are still working on that. Now, the morbidity and the mortality that come from malaria come from different things. These people have severe anemia. So I'm a hematologist, oncologist. We see patients in our clinic, and we think they have severe anemia if their hemoglobin is maybe 9 or 8 instead of the 12 or 13. In these children and some of these adults, their hemoglobin, instead of being 12 or 13, is maybe 5 or 4 or 3. They are severely anemic. And that's part of the definition of severe anemia. The hemoglobin has to be less than 5. Some of the people die simply from anemia. They can't get enough oxygen to their tissues, so it's a serious problem. Cerebral malaria is characterized by grand mal seizures and coma. It occurs gradually. It's related to malaria, the parasitized red cells adhering to vasculature and blocking blood flow. It's a very complex thing, but many patients die from cerebral malaria. There's also an instance of hypoglycemia, that is a low blood sugar, of renal failure. This is more so in adults than children. Lactic acidosis, acute respiratory distress syndrome with pulmonary problems. And also these people, when they have malaria, have a high chance of developing non-malaria infections like salmonella. So it's a very bad disease. So what is it that causes the symptoms and the signs and the severity of the malaria? For years, people have looked for, quote, the single malaria toxin. What is it that's making these people so sick? In the 50s and 60s, there was a lot of research, and people thought that it was related to endotoxin or lipopolysaccharide that came from something. We usually think of microorganisms like bacteria. They were thinking it was coming from the parasite. Also, tumor necrosis factor, interleukin-6, macrophage migration inhibition factor, secreted by macrophages, hemozoin, so the degradation product of hemoglobin that the malaria parasite digests, the glycophosphatidylanosytol inositol that comes from the membrane of the parasites. All of these have been talked about and published about as being related to the toxicity that people have when they have malaria. And then in the 70s or so, as nitric oxide biology became more important, several people stated that they thought that nitric oxide was the malaria toxin. So nitric oxide does a lot of things. We all have nitric oxide in our bodies. Several of our drugs are prodrugs for nitric oxide, like nitroglycerin is converted to nitric oxide. And it relaxes blood vessel musculature, decreases blood pressure. It also serves as a neurotransmitter. It can be a toxin for the brain, etc. There were several people who wrote individual papers and reviews about nitric oxide being the malaria toxin with zero data. They had not measured a thing. So when we set out to do this work, we wanted to know if nitric oxide really was the toxin. In other work, so I worked with nitric oxide in leukemia and hematopoiesis and inflammation like an arthritis. Dr. Granger worked in infectious disease with fungal diseases, et cetera. So we were all set up to measure nitric oxide. So in 1993, 94, when Dr. Anstey was a fellow here in infectious disease, he, with the help of Dr. John Hamilton, wanted to determine if nitric oxide was the, quote, malaria toxin. So this arrangement with people in Tanzania was that he and his wife, with their first son was 2 or 3 weeks old when they moved to Dar es Salaam, Tanzania for this project, wanted to study this by studying children with malaria. Sort of uncomplicated malaria and severe anemia, cerebral malaria. Actually, in our first cohort, every patient had cerebral malaria. And what we found was that this is sort of a summary of the talk here, so pay attention. Nitric oxide is not the malaria toxin. We found that if the patient wasn't able to make enough nitric oxide, they were more apt to develop severe malaria. And if they could make a lot of nitric oxide, they did not develop symptoms of disease, even if they were infected. And nitric oxide was protective against severe malaria. And it was not directly anti-parasitic if we did the quantitation of how much nitric oxide they were producing and how many parasitized red cells and how much parasite you could find in the body. And we at that time weren't thinking too much about endothelium, but over the years we found that nitric oxide corrects or reduces endothelial dysfunction. Now, one of the central things in the pathogenesis of malaria is that the parasitized red cell expresses parasite proteins in the red cell membrane. And these parasite proteins will then bind to cell adhesion molecules on endothelial cells. and then modify endothelial function, and sometimes clump enough that it actually blocks the blood flow through the blood vessels. And so there's ischemia. And NO will inhibit this binding of parasitized red cells, endothelial cells. And so we think, and we still push that, we think increasing nitric oxide might be a good way to provide adjuvant treatment. You still got to use antimicrobials, anti-parasitic drugs. But as I said, even if you do your best, these patients still have severe disease and many of them die, so we want to look for something better. So what I'm going to talk about is causes of low nitric oxide in malaria, the mechanism by which this low nitric oxide level and production takes place, characterizing and quantifying endothelial dysfunction in malaria. Talk about a new thing for us, and this will be some unpublished data, the endothelial glycocalyx. I'll explain what that is in a minute, and how you can actually measure that. And then finally talk about some possible adjuvant treatments for malaria. Now, nitric oxide, also called nitrogen monoxide, is NO, one nitrogen, one oxygen. It's a reactive free radical. It can act as a neurotransmitter. It modulates vascular tone by controlling smooth muscle relaxation that's in the blood vessels. And it can modulate inflammation. With some microbes, like certain fungi and certain bacteria, nitric oxide is important for resistance to those infections, especially like tuberculosis and fungal infections. And there are 3 genes that encode for enzymes that make nitric oxide from the substrate arginine. So nitric oxide synthase 1, 2, and 3. These are results from the first study that we did. It's supposed to be a reference there. It's not showing up. This was published in 1996, showing that the level of the nitric oxide synthase type 2, the inducible nitric oxide synthase, the level of nitric oxide in the blood was relatively high in healthy control individuals and those who had parasites in their blood but no symptoms of parasitemia. They were infected, but they were controlling it and they didn't even know it. We recruited them as controls, but we found the parasites, so it's called asymptomatic parasitemia. So their levels were quite high. However, in patients who were sick, uncomplicated malaria, fever, feeling horrible, some of them needing to be hospitalized, some of them not, we treated them with anti-parasitic medicines. And in the most severe cerebral malarias, these were extremely low. So if you didn't make enough, you didn't have enough on board, you really got sick. And this was published here in the Journal of Experimental Medicine, and take note of the year. So this was 21 years ago, almost 22 years ago. And so right away, we're saying nitric oxide is not the toxin. Now, nitric oxide, as I've just shown you, is such that there's an inverse relationship to disease severity. We first showed it in Tanzania and then Gabon at the Albert Schweitzer Hospital there. And then later in Indonesia, Dr. Anstey moved back to northern Australia near Indonesia. And high nitric oxide and NOS expression is associated with no disease at all or very mild disease. And if it's low, you have severe disease. We didn't know why any of these things were happening. This is a reaction. So arginine is a simple amino acid. We all have it in our bodies. We get it with our diets, etc. The enzyme nitric oxide synthase converts arginine to nitric oxide and citrulline. And the story is much more complicated than it seems, and I'll explain that. One of the findings that was made by Dr. Bert LePandry here, he at that time was at Salt Lake City, Utah with Dr. Granger, had moved from Duke to there. He was a fellow, and now he's head of infectious disease at their pediatric hospital. He showed that with us that in healthy controlled children, you have a certain level of arginine, the substrate for this enzyme. And it's about 120 or so, 125, 130 in American children. It's about the same in Tanzanian children, too. But if you had uncomplicated malaria, it was lower, significantly lower. If you had severe malaria, it was even lower. And the black dots and the white dots here are meant to represent those patients who lived, the black, are those who died with cerebral malaria. And it was shown here that low arginine was directly related to the chance for death. If you had the lowest arginine, you were more likely going to die. And these are children age about 1.5 to 5. Now, arginine is taken in from the diet, but it's also synthesized. There's a de novo synthesis here. Most of this takes place in the kidney from glutamic acid, glutamine, glutamic acid, citrulline, and then converted to arginine. And we've done a lot of work, and I'm presenting this sort of out of sequence of when the work was done. We eventually analyzed, with the help of a lot of people here in Utah, Dr. Millington, who's here today, and his workers in pediatric metabolism, to say, well, is there some problem with being able to make the substrate? Is that the reason it's low? And so we measured everything along this pathway. Like, here's the pathway here. I had to leave out a bunch of slides, but here's the pathway to get to arginine. So glutamine in uncomplicated malaria, cerebral malaria, or severe malaria, low, significantly low, every one of these. Glutamate, very low, extremely low. And these were a lot of patients here. Citrulline production, extremely low. Arginine concentration, I've shown you this, more of the same. And then ornithine was also low. So there may be some sort of problem with synthesizing just in our body to make enough arginine. It's complicated because at the same time it can be synthesized, arginine can be broken down. It can be metabolized by this enzyme called arginase. Arginase can be made by monocytes and macrophages, a lot of different cells in the body. This is a complicated slide, and I won't go through everything. Here's that reaction again, arginine converted to nitric oxide by nitric oxide synthase. We've already shown that these precursors for arginine are low in malaria, but at the same time, arginase, and work I won't give you details on, in malaria is increased in the plasma and in the tissue fluids. So arginase converts arginine to ornithine, and you basically get rid of arginine, so it's another way you can have low arginine. And then up here, we did complicated studies. Each one of these I could give a lecture on, but I'll hold myself back from that today. The NOS gene is there. We've discovered certain polymorphisms in the promoter region of these enzymes so that there's altered production of transcription of the gene. So the messenger RNA is low in these patients. We still don't know exactly why. Some of it's due to polymorphisms and some other things. The protein is low, determined by immunoblots. And then the actual enzyme activity in malaria is low. So NOS is low too. So arginine's low, NOS is low. I'm going to point out on this figure, everything you can measure that would influence the ability to make nitric oxide in people with malaria is low. It's just wiped out. Also, once you have the enzyme there, you make a little bit, you've got to have these cofactors. And one of the very important ones is tetrahydrobiopterin. Complex chemistry there, but it's necessary for this enzyme to work. And we demonstrated that this is low in both adults in Indonesia with malaria and children in Tanzania with malaria. And then also there's some inhibitors of the enzyme that compete for arginine for this enzyme. I won't go into detail here either, but they're modifications of arginine, like asymmetric dimethylarginine. They're very high in malaria. And it interferes with whatever enzyme activity might be there. And then finally, if you do squeeze out and make just a little bit of nitric oxide that might be doing something good, since in this disease, the part of the infection is the parasite gets in the red cells, and then the red cells are destroyed early, and as they rupture, they release hemoglobin into the plasma free. And hemoglobin will actually bind nitric oxide and inhibits its bioactivity. It's a quencher. So another thing that's bad for having functional activity of nitric oxide. So we're messing up in every step here. We've worked a lot in this. I won't go through all of these, but just to summarize sort of, there's low overall nitric oxide production. There's low messenger RNA for the enzyme and the protein, and when we measure the enzyme activity, it's very low in malaria. There are promoter polymorphisms that modify nitric oxide production by the enzyme. The cofactor biopterin is low. A lot of this work, by the way, is done by Dr. Matt Rubach, who's now faculty here in infectious disease. He's based mainly in Tanzania. There's low substrate. That's the arginine story. That was established back in 2003. And then every time we do a study, we have to measure arginine, and it's always low. In any person who has malaria, it's always low, both adults and children. There's high arginase activity that destroys whatever arginine you might have. And then there are these endogenous inhibitors of the arginine, of the nitric oxide synthase, and they modify arginine transport. And then there's an increased free hemoglobin, which acts as a nitric oxide quencher. So we got lots of problems here. Now let's talk about the endothelial function. I mentioned that important in the pathophysiology of malaria is the parasitized red cell, somehow or another expressing parasite proteins in the membrane. It's an automatic almost. And then these proteins act as ligands for receptors or adhesion molecules on endothelial cells. So you can aggregate the red cells, or you can modify the endothelial function further. So they do bind there. And activated endothelial cells have the increased cell adhesion molecule expression. And we didn't make the original observation. A lot of people noticed in the patient with malaria, there are a lot of cytokines that are high. Important there is tumor necrosis factor. And TNF is one cytokine that causes increased cell adhesion molecule expression in endothelial cells. So once again, that cell is going to be binding there. They do bind by these cell adhesion molecules. And for example, it would be like intracellular cell adhesion molecule type 1, sometimes called ICAM1, vascular cell adhesion molecule, ECAM, etc. And then it's sort of a vicious cycle. Once these things bind to the endothelial cells, just as a response to that modification, the endothelial cells start expressing more of these adhesion receptors. So you get more and more adhesion and more pathology there. And then, as I say, it's sort of a vicious cycle. And importantly for our story is if you've got enough nitric oxide being produced either by the endothelial cell or by other cells floating through the blood, nitric oxide blocks or inhibits all of these bad things. So if you don't have nitric oxide there, they're going to be worse. And we think that's important to the nitric oxide story. So how could we measure endothelial function? All of our work has been done in humans. There are mouse models of malaria. So malaria infection is very species specific. So there are 5 main species that infect humans. And then there are some species that infect mice, some monkeys, apes, etc., some birds. So a lot of investigators, including me, have done a lot of work with mice. So all my leukemia work, we've done mouse work. It's very convenient because you can get animals that are inbred so they all work alike. You know, like mouse 1 is like mouse 3, is like mouse 7, etc. So you don't have to worry too much about genetics. But as far as the malaria story, you can have parasites that infect the mice and they cause disease. They get parasitemia. Many of them eventually die. The cerebral malaria story is different. The red cells that are infected with the parasites in mice do not bind to the endothelium well. So it's a different physiology. There is a, quote, cerebral malaria model in mice, but it doesn't look like human cerebral malaria. And so we've stayed away from that, although there are some excellent researchers who use mice in their work, and they have derived a lot of important information. A lot of the story summarized about nitric oxide has been confirmed in mice. This is one of the unusual things where it was first done in humans, and then the mice people went back and they found about the same thing, almost exactly the same thing we did using mice. And so they're excellent mouse researchers, but we stayed away with that. In measuring the endothelial function, we wanted to do this in humans. So there is one thing that we do called measuring reactive hyperemia using peripheral artery tonometry, or HPAT. And what happens here is if one of us had a tourniquet put on and you elevate the pressure in that so that you essentially block arterial blood flow to your arm. And you can do that. It's a little uncomfortable, but you can stand it. And so like, we do that for 5 minutes. And then all at the same time, we're monitoring the pulse waves of the blood that's flowing through the arteries. We have something that I'll show you in a minute we do that with. Also, it can be done with ultrasound where you measure the diameter of the brachial artery. That's harder to do. So when you release it, the blood just gushes down there. And there's a certain shear force over the endothelium. And it changes things a lot. One of the things it does is it causes a lot of nitric oxide release from the endothelium. And that nitric oxide dilates the artery, and you'll get this reactive hyperemia, increased blood flow. We bought one of these. We bought several of these machines, actually. This company calls it the Endopat device, and it measures this flow-mediated vessel dilation. There's a blood pressure cuff. We actually have one on both arms when we do it. There are these finger probes down here that measure the pulse waves, and you can quantify the hyperemia that occurs, either the presence or the absence of that. These are some tracings. This is taken from one of the guys who did it first in cardiology. Cardiologists are all over this, as you might imagine. And what we see here is the tracing. So here it goes along. Each little blip is the beat of the heart there. And then when you put the tourniquet on right here, so these people have the tourniquet placed. We usually do both arms. So on one arm, we inflate the tourniquet, we inflate the blood pressure cuff so that there's no blood flow during that time, and the other one, we just leave it alone. And this is what we see in a normal person here. There's no blood flow, and then when you release it, notice how the amplitude of that is much higher. And so that's compared to someone who might be abnormal. Well, there's the nothing. And then here, you compare this to this and quantify how much that is. This is a person, this was from this heart journal here, this was a person with severe atherosclerosis. So this person had no hyperemia after the occlusion. Notice this is just the same as that. So these are measured, and you can do measurements on that. And so we did this. Actually, this was done by Dr. Sen Yo, who originally is from Singapore. He worked at WashU for residency, or maybe Utah, and then IB fellowship at Utah, and then now is back in Indonesia and Australia and back in Singapore. But these studies were done in Indonesia and showed that there was low nitric oxide, but also endothelial dysfunction and malaria. This is a picture of Dr. Yo and some of the investigators from Indonesia, Dr. Lampa and Dr. Gidawati, who did these studies. This was at a tropical medicine meeting where this data was first presented. And the findings were that patients with severe malaria had very defective reactive hyperemia using that technique that I just showed. They also had very low nitric oxide production, and they measured that in the blood. And then you can also measure nitric oxide with a device where when people exhale their breath, you can measure nitric oxide in that. So the people had low nitric oxide. And data I won't show you, in the same group of patients, they had permission to infuse arginine in these people, and the people had low arginine levels also. And so we infused a lot of arginine, grams and grams of it, and there was a dose response, so you could elevate the arginine levels back to normal, and when they were elevated, the nitric oxide production normalized. There was no toxicity. The arginine had an extremely short half-life in people with malaria compared to normals. And we feel like that's partly due to the increased arginase. And it corrected this endothelial dysfunction. And work I won't talk about, we later showed that in a trial to see if just giving people arginine would help them, would make the malaria less severe in the long run. And what we showed was that the half-life of the arginine we infused was so short that we just couldn't keep the level up. And we think it's being destroyed rapidly, so we don't think that's going to be a good treatment. Now, as I mentioned here, I showed this slide before, arginine is very low in patients with malaria. We didn't know why it was low. I've already told you it may be from production or maybe from destruction. Like here, maybe you get some arginine synthesized down this pathway, and arginase at the same time is just destroying it so fast you can't keep up with it. And or maybe there's a lot of nitric oxide being made. We already know that's not the case. And in some complicated studies, we measure this with the help of Dr. Millington and his coworkers and pediatric metabolism here, using labeled arginine, so non-radioactive label of arginine, to do flux studies to talk about the turnover of arginine to see if it was really increased. And I won't show you all the pure data on that either. I'll just summarize it by saying, as I've already shown you, the arginine precursors, amino acids that are used to build arginine, are extremely low in patients with malaria. And overall, there was no difference in arginine flux, that is the net of arginine production and disposal, when we compared healthy children in malaria and children with malaria. These were hard studies to do. We did them in Tanzania. So we went there and we recruited children from age about 4 to 7. The parents were all involved in this. So if you have children, or when you were a 4-year-old, would you like to be lying in bed for several hours to have this infused and then multiple blood draws? They were anxious to do it because they realized how severe malaria can be. They wanted to do anything. So we finished this with just short of 20 patients, or 20 healthies, a total of 20 people. And so the general thing was that there wasn't a difference. We worried that maybe the people with malaria at this time didn't have really severe malaria, because we weren't seeing as many people with very severe malaria. And the other was that maybe we need a higher sample size. We had about 7 or 8 healthies and about 9 or 10 people with malaria. But anyway, we did not see an obvious change in the flux. Next, I want to end by talking about the glycocalyx. Most of our cells in the body have this glycocalyx. We were interested in the glycocalyx of the blood vessels. So this is a gel-like layer that's composed of these compounds, glycosaminoglycans, and certain glycoproteins, and it can be up to 5 millimeters thick. Now, if you've ever looked at blood under a microscope or seen pictures of blood, a red cell is about 7 to 9 millimeters in diameter. So this is a pretty big layer, and it's inside the blood vessel, even the small blood vessels. And it's on the luminal side, the part where the blood flows, of the blood vessel, of the endothelium, and it acts as kind of a protective barrier. So glyco, sugar, then calyx, meaning a husk or a shell. There are proteins involved like syndicans, glypicans, and then these glycosaminoglycans, heparan sulfate, dermatan sulfate, chondroitin sulfate, hyaluronic acid. And some believe that impairment of this barrier of the glycocalyx may be the first step in gross microvascular dysfunction. If your blood vessel function is poor, endothelial function is poor, they think that if you're messing up your glycocalyx, it's going to have a big effect. And in addition to just being sort of this slick barrier, it does act as something where certain proteins will kind of slip in there, and it acts as kind of a reservoir for certain proteins like cytokines and other proteins. And it does act as a mechanotransducer for shear stress regarding the flowing blood or the endothelial cells And I'll show you how this pressure on this sort of squishy glycocalyx can modify the function of the cells underlying it. And one of the things that's been demonstrated extremely well is that as there's shear stress over that, it actually induces nitric oxide production by endothelial cells. This is a figure from someone. So these would be endothelial cells where there can be some protein sticking out, like these syndicans and glypicans. And then up in here, these long things can be these glycosaminoglycans. And the example here is heparan sulfate, chondroitin sulfate, hyaluronic acid. And some investigators sacrificed animals. I think these were rats or guinea pigs, and they used some special fixation techniques so that this glycocalyx part would be intact, and you can see what we're talking about, this fuzzy type stuff. This is a close-up. This is like the endothelial cells here, and this is the glycocalyx sort of wiggling there. You don't think about that until, I didn't at all until about a year and a half ago when I started studying this. So it's in all of us. This can modulate vascular permeability. It moderates access of cells to the endothelium, like neutrophils and lymphocytes migrating through. It can regulate cell adhesion molecule expression. Of course, that's important for us if you're thinking about these parasitized red cells binding. And it can mediate signals from shear stress to enhance this vascular growth factor and nitric oxide production. This is an example from this paper here in 2003, where we're measuring nitric oxide levels over endothelial cells over time, and they're flowing things over this, so there's shear stress on it. And in the controls, there's very low nitric oxide production, the green here. In those where the shear stress is increased by flow, there's definitely an increased amount of nitric oxide production. If you first treat those endothelial cells with heparinase, an enzyme that will take away certain glycosaminoglycans, the control is nothing happens. But in the same cells that you did the flow over, here in this purple, you abolish the ability to make nitric oxide, just demonstrating how important the glycocalyx can be for transducing pressure signals. Now, in assessing the glycocalyx, you can image the glycocalyx in living people. I didn't know this was possible, but I learned about it. There are certain cameras where you can actually see the blood vessels. I'll show some of that. And you can also quantify glycocalyx breakdown products like these glycosaminoglycans. This can be done in colorimetric assays and by mass spectroscopy. This is a picture of this camera that we have. It sort of looks like this, and there's this probe. This lady has it sticking under her tongue, the sublingual area. So there's a camera there, and I'll show you. You actually see the blood vessels sort of in relief, because what you're seeing is the red cells flowing through the blood vessels in real time. And you can quantify this. And here in this figure, healthy glycocalyx, the red cells are sort of confined to this region. And there is this perfused boundary region where some of them will be here, and it's all kept away from the endothelium right here by the glycocalyx in the dark blue. But if something's damaged in the glycocalyx, the red cell column and the blood flow column will get out and get closer to the endothelium. This is being used in other disciplines. This is a paper that's published this last month, studying patients who have systemic sclerosis. This is also known as scleroderma, where there are a lot of vascular abnormalities. I won't go into details, but in that, they measured a lot of things using the same camera that we've used. And what we're doing is a sort of a diagram like the last one, is once this is captured by the computer, there are computer programs that automatically measure things here, and you can determine these parameters. And the one thing is that if the glycocalyx is damaged, this perfused boundary region is increased. And so this is one of the key parameters that we're looking for. You get a lot of measurements out of this. This is all done in the background. You don't have to measure anything with a ruler or anything. It's done automatically, and they saved images. And we've done that. Studies done before in healthy, aged people, septic shock survivors, septic shock non-survivors show that this PDR is increased. And this is a picture here showing, if you look, you'll see the red cells going through these blood vessels. You see that? That's what it looks like on the camera when we do this in our patients. I'll do that again. Oops. So you see these, and this film is captured and then everything is measured automatically. This is Dr. Salvatore Florence doing one of our patients in Tanzania. This was a healthy control, excuse me. And then he has his probe in the sublingual region. And the next slide, his buddy was taking pictures of the screen at the same time so he can see in real time. Not quite as stable as the other one, but you can see the red cells flowing through that. So all these measurements are being made, and we're collecting this to try to correlate nitric oxide parameters that we measure, degree of severity of malaria. So we're doing this in healthy individuals and patients with malaria. And yes?
I have a question. So blood vessels, they have different diameters. How can you kind of compare control and patient?
They actually measure the diameter. That's one of the parameters that comes out. And they're not large vessels, they're small vessels.
Just small vessels.
Yeah. So I think they go up to 100 micrometers, but the size that is most revealing is the 5 to 25 micrometer diameter vessels. So they're pretty small. That's a good question. So in general, the glycocalyx is the protective coating of the vessel wall. You can measure this non-invasively. The damage increases with age, just older you get, and that was true with all our volunteers. You know, the younger students and people working in the lab had a value that was pretty small. 50-year-olds were much larger. The 73-year-olds were quite large. And these people were apparently healthy, but it did relate to age, but also diabetes, cardiovascular disease. Some papers have been published about bacterial sepsis. And in general, we've done that. I'll show you some of the results. In addition to that, you can get data that I think is more quantitative. That is, you can measure the release of these glycosaminoglycans from the damaged glycocalyx. And there's a colorimetric assay where we add this dye that will bind to sulfated glycosaminoglycans. It's like a microtiter plate assay, if any of you ever worked in the lab. So the 6 millimeter diameter wells that hold like 0.2 ml. You can put a sample in there. We measure urine where these things are excreted. You can put the sample in there and then add this reagent, and instantly you'll get a color. It's really dark blue if there's a lot there, and there's no color or very light blue if there's not much there. And that's it. You know, we can do that assay. And then we have automatic plate readers to measure the density. More difficult but more accurate are mass spectroscopic techniques, more complicated than the same path, where we can measure individual ones. And we focused on chondroitin sulfate, dermatan sulfate, and heparin sulfate. These are very accurate. They're relatively expensive. These are cheap. These are more expensive. And they're time-consuming, but we feel like you get a lot more information because you can actually look at different ones, and there may be some differences in different organs of the content of those. And these are some results from the original urines that we collected in 1994 and 95. One of the advantages to these chemicals, these compounds, is they're extremely stable. So these were collected from the sick children and well children in 1994 and 95. We saved them, so you got to save all these samples for this research. Don't throw anything away because we'll go back and look at it. And here, notice it's a log scale, so 0.11, 10, 100 grams, moles per creatinine. We base this on urine creatinines. So in the healthies, there's a pretty wide range here. But clearly, in those people with mild malaria or uncomplicated malaria and cerebral malaria, it's higher. And when we go back and look at all the great details of those patients we had at that time, like how many parasitized red cells did they have per microliter, how sick were they, et cetera. These results correlate extremely well with how sick they were. So it's a good correlation with degree of sickness. We did the more detailed studies, and once again, this is a log scale. We can't figure out why this one is so low, but that's just the truth there. For chondroitin sulfate, once again, this was quite significantly elevated in uncomplicated malaria and in cerebral malaria. And here we just lumped all these together. All of these plus these. So these are definitely increased. And in general, this is what we wanted to show. This was our preconceived notion. They were done blindly, so no one, when they were doing the assays, knew what the patients had. But it showed what we thought it would. That is, chondroitin sulfate was increased. There was disturbance of the glycocalyx. Likewise, heparan sulfate was increased. The data is a little more tight here. These are clearly very significant. And the other one is dermatan sulfate. Dermatan sulfate's interesting. The glycosaminoglycan content in the glycocalyx in different organs, in different vessels from different organs, is not totally known, but it is known that dermatan sulfate is relatively low in the brain microvasculature. So this kind of fits that it's not disturbed. And then we've also done this in adults, so those are all children's studies. We've done this in adults from Indonesia. This is from Papua. These are urines that correlated with that Journal of Experimental Medicine paper done in 2007. And once again, these are definitely high in patients with malaria. Here we do two species. Over there, we have a lot of Plasmodium falciparum, PF, and a lot of Plasmodium vivax, another type of malaria. And it's elevated in both of those. Chondroitin sulfate, elevated again. And then heparan sulfate, elevated. Now, there's some minutiae that I won't get into here, but these were definitely elevated. And once again, the dermatan sulfate was not elevated in these patients. So there is difference in the different glycosaminoglycans. Now, the other place that we've done a lot of work is in Malaysia. We, meaning Dr. Sin Yo and Dr. Nick Ansby, have led these studies over there. And the story is the same. And there they have the advantage of looking at a different species of malaria. This is Plasmodium knowlesi. It's one that's been discovered in the last 5 to 10 years. This is the one that they believe even now is going on. Infection being spread from primates in the jungle or in the woods directly to humans from mosquito bites. And it is a severe malaria. So I could go on and on here. There's some other things we found is that once these are elevated, like when they present to the hospital, they're high. And if we measure it 24 hours later, 48 hours later, it stays high. A lot of the things in malaria, once you start treating them, their disease resolves rapidly. So they're getting better, but these things remain high. So we've done some correlation studies, and the statistics on this have not been finished, but the glycosaminoglycans, the height of the glycosaminoglycan abnormality is correlated with the flow-mediated dilation defect. So the worse, the higher the glycosaminoglycans, the worse their flow-mediated dilation is. And also, the level of abnormality of glycosaminoglycan relates to the level of parasitemia and also a measure of malaria density, measuring a certain protein that's elaborated by the parasites, the histidine-rich protein type 2, HSPR2, and we've done those too. And then there weren't many deaths in the groups from Indonesia, but in those who died, doing statistics on relatively low numbers, it was clear that the ones that had the high glycosaminoglycan excretion were more likely to die. So it seemed to be a predictor of death, of severity too. I won't go over this too much, but these are elevated in both children and adults. So children, The biggest problem as far as deaths and all, but adults in some regions where you have people who do not have immunity moving into areas where there are a lot of mosquitoes and malaria transmission, they have no immunity, they can get extremely ill. And there are differences in childhood and adult malaria. And it's seen in 3 species. It's elevated on admission and up to 3 days after admission and after treatment. And these correlate with disease severity and many other things. We've done some studies actually from Dr. Taylor in his lab where we've looked at parasites growing in vitro in tissue culture media. And we showed there that there's no evidence of increased glycosaminoglycan in these cultures. So one possibility was all the stuff we're measuring might just be coming from the parasite and not the endothelial cell. But we have no evidence that these parasites will make these glycosaminoglycans that we're measuring. There's a lot more that we need to do. We're still analyzing the data. We're excited about this because it's never been done before in patients with malaria. It's never been done before in children. So just in summary then, activated endothelial cells have increased cell adhesion molecule expression. They also have endothelial glycocalyx damage, and the parasites will bind to the red cells. Parasites in the red cells will bind to the endothelial cells, and this binding induces more cell adhesion molecule expression. Is there a way to repair this or to treat people? A lot of things have been tried. Most of them don't relate to the diseases we're looking at. Antioxidants have been used, statins, like are used to reduce cholesterol. There are some studies where people will take statins and these things are measured, and they're measured after 5 months of treatment or something. Acutely, they can't show anything, and they're not impressive. Fresh frozen plasma, just feeding or giving IV these glycosaminoglycans themselves, the data is very poor there, and most of it's done in rats. Exercise may have an effect, but it's an exercise program existing over months and months. There's one recent paper showing that fibroblast growth factor may be something that will acutely change the glycocalyx and repair a damaged endothelial glycocalyx. This is a paper related to that from the pulmonary doctors in Denver, and we've been communicating with these people, and they actually show that this fibroblast growth factor in vitro systems will repair damaged glycocalyx. So it's been shown in a few other situations that there can be glycocalyx damage. Bacterial sepsis, septic shock, renal failure, dengue is the most recent disease that might be close to what we're looking at. Acute respiratory distress syndrome, the people at Denver have studied this. The man leading these studies is a pulmonologist and an intensive care unit doctor, and then a few other. So the implication here is that this glycocalyx is probably important for normal function. I think that's not debated at all. And we've shown that the glycocalyx is abnormal in malaria, and either preventing it or repairing the damaged glycocalyx might be a new target for adjunctive treatment in malaria. Now, over all of this work, we keep thinking, is our work going to lead to some new treatment or something? And I'll just flip through this. First of all, treat malaria, you have to use conventional antimalarial antibiotics. Secondly, you could increase the substrate. We tried that, and that didn't work with arginine. It was low. You could inhibit arginase. This has not been done methodically. There are a few things that will inhibit this enzyme. None of them are approved for human use. You can increase NOS enzyme activity and NO production by giving certain cytokines like M-interferon, alpha-interferon, endotoxin. Not been tried. You could reduce inhibitors. This is not possible as far as we know. You could decrease NO consumption in the way of lysis. Once you're infected, you get lysis of the red cells going on almost all the time, so we try to reduce the malaria there. And you could provide more reduced terrans. The sepiaterin is something that has been given to humans in certain genetic disorders, so that's a possibility. Some groups are looking at that. You could administer more NO. This has been tried with NO, which comes as a gas. There are a couple of studies that have been published, and it did not work. The problem with those studies, we feel, is that it's given by inhalation. It doesn't get to all parts of the body well enough. And then you could change monocyte function so that they don't produce as much arginase and they have more nitric oxide synthase. That's hard to do. And then you could work on the glycocalyx. These are the ones that I think are reasonable, and these are being explored by us and by others too. So this is the first slide I showed. I've covered the areas and I'm about out of time, so I guess I'll just say thanks to everybody. Thanks to you all for listening, and I'm happy to answer any questions if you have some.